Hi, I'm Kieran H. DeWitz, and I'm going to be describing to you the anatomy and the function of a neuron. So let's start at the top with dendrites, which receive information from other neurons, and they're these little pink tentacle-like things over there at the top. So that's where the neurotransmitters will head, and there are sensors on top of the dendrites that will receive the neurotransmitters, and then the neuron can do whatever it wants with this information. Then below you'll see this purple dot there. That's the nucleus. That's the center of the neuron that's responsible for its function, like the control center. It stores DNA, it makes proteins, and all kinds of other good stuff. It's stored inside the soma, so that's a cell body because it's the body, like the interior of the cell. So that's where the nucleus and other structures are contained within the neuron. So then this big part, the axon, it's that whole length. It's like that big long part, and so it's a long bundle of fibers that carry information away from this neuron. So information is like passed along the axon, and so this is obviously a pretty important part, just like all the others, and it's really important. So it has protection. So the myelin sheath is this sort of tube-like thing that's a fatty insulator and protective covering, but it does more than just like protect and cover. It also speeds up the transmission and aids in the function of the neuron. So this myelin sheath, not only will it be unprotected, but it won't work. So without this, without any of these pieces, really, you won't have functioning neurons, which means you can't really move. You can't receive signals, send signals. So neurons are very important. So is the myelin sheath. Without it, if it's degraded, you can't touch, feel, move. So the Schwann cell now, that's what forms the myelin sheath for neurons outside the brain. So that's an important distinction. So inside the brain, there are glial cells, which are creating several myelin sheaths, but Schwann cells just create one, and of course, an important part of the neuron. So another bit that's interesting to note is the node of Ranvier. So you'll see that there are gaps in the myelin sheath where it just sort of ends and leaves a bit exposed. So that's what helps promote action potential. We'll talk about that afterward, which is the positive sweep that causes the neurons to fire. The axon terminal is all that bit down there, all those like little tentacly pieces and stuff. That's the axon terminal where the neurotransmitters are stored, and that's where they're fired off from, those pieces. That axon terminal is what extends outward, and that's what can be really close to the dendrites of another neuron. So more specifically, the closeness is a synapse. So that's the space between the axon terminal of this neuron that we're talking about, and the dendrites, the top of another neuron. So the neurotransmitters are fired off of the axon terminal of this one, travel through the synapse and onto the dendrites where there are sensors for the neurotransmitters, and that's what causes something to happen, whether it's like receiving information, giving information, whatever it is. So yeah, that's the anatomy of the neuron, but now we can take a closer look at how it works. So inside and outside the axon, there's different environments. So here we have Na+, positive ions, inside and outside, but obviously there are way more outside. That's because the axon is pumping Na+ outside and creating a positive exterior environment, negative interior environment. So this whole environment is resting potential. But then once the membrane becomes permeable, that's when all of the Na+ wants to be in the negative environment, because positive and positive, negative and negative, it doesn't work, and it wants to be mixed. And so then all this rushing in of the positive, that's that crashing in is permeability, and this positive wave of Na+, of positive ions, rushing in is the action potential. So this sweep down the length of the neuron is the firing of the neurotransmitters. So then you'll see the neurotransmitters in blue there. They'll be fired off because of this action potential positive sweep, and then they'll be sent over to another neuron, to the dendrites, and they'll be sensed, and then something happen, maybe it'll get passed on even more. So this is happening within milliseconds, so fast, so many times, but after this firing, it needs to rest. So you'll see the refractory period, the resting time after the firing, because it can't just like fire. It has to build up that resting potential. It has to build up that positive exterior, negative interior environment. So that's the refractory period right here, just like resting, relaxing, and then of course the neurotransmitters have all been like fired out, or like in the synapse or something. But then it'll have to be recollected, and the reuptake is when the neurotransmitters are taken back into the axon terminal of the neuron. in order to stay there and wait for the next firing. So that's the resting period, the second one. And so after that, it's like resting, as you can see with the Z's, and it's taking a break, and then the whole cycle can begin again with the resting potential, permeability, action potential, all that good stuff. So that's the function of the neuron, and I hope you enjoyed, and thank you for watching.